Hello everyone, myself Manju Sharma from Suresh Kanviar University, Jagatpura, Jaipur, and department is School of Agriculture. Today we are going to start diseases of banana. Diseases of banana. Basically, today we are talking about major diseases of banana, in which the first disease which we are going to discuss, that is Shigatoka disease. So first of all, their economic impact. Yellow Shigatoka, that is worldwide in distribution, first observed in Java in 1902 and it epidemic in 1913 in Shigatoka Valley in Fiji. That means in 1902, basically it was observed only in that was worldwide distribution, but it was epidemic during the 1913 in Shigatoka Valley in Fiji. That's why the name given Shigatoka disease. Okay, because it was epidemic in Shigatoka. That's why not reported from Canary Island, Egypt, and Israel. In India, yellow shigatoka is a serious threat to banana production in the states of Assam, Tamil Nadu, Karnataka, and Andhra Pradesh. Black shigatoka is not prevalent in India. Now, their etiology, yellow shigatoka, which goes by the Mycosphaerella musicola. Yellow shigatoka, that is goes by Mycosphaerella musicola and Pseudosarcospora muci. Black shigatoka or black leaf streak, which goes by Mycosphaerella physinensis and Parasarcospora physinensis. Pathogen. Infection takes place through ascospores in the infected plant debris. Infection readily takes place through stomata on the lower surface of young leaves. So the disease is spread through wind-borne conidia. Surface moisture is necessary for release of both conidia and ascospores. Hence, the disease is severe in moist weather because that is also a fungus. So, whenever the weather will be moist or more humidity will be there, in that case, the fungal disease will be more or the fungus which can grow very fast. And here you can see the conidia of Pseudosarcospora muci. That is the life cycle, like how the fungus which will be cause the disease. So first of all, like there are the ascospore or pseudothecium. Then firstly they will attach the surface. Then ascospores are wind dispersed over long distance to new plant. Like through the wind, there will be transmission of the pathogen. After that, ascospore infect leaves through the stomata. Then infection produce thin brownish streak that darken and expand into large areas of necrotic tissue. After that, mature lesions produce sexual structure and after that they will be converted into spermatogonium in which the spermatia will be there. Then the spermagonia fertilize neighboring reproductive hyphae to produce the pseudothecia, and ultimately the conidia infect the leaves throughout the stomata, and the conidiophore emerge throughout stomata and produce conidia. So like that there will be the life cycle of the fungus. Favorable conditions like high humidity, heavy dew, and rainy weather with temperature above 21 degrees centigrade, prolonged leaf wetness periods, poor drainage, and low soil fertility, particularly of potassium. Then closer planting, because if the planting will be closer, then more humidity will be there or more moisture will be there. So if moisture will be there, then the fungus which can easily grow there. Poor drainage and low fertility, particularly of potassium. Closer planting, susceptible cultivar like Grand Nene, dwarf, dwarf Cavendish, and giant Cavendish, and more suckers in emmet because of non-removal. So these all are the favorable condition for the pathogen to cause the disease. If all the condition will be there, then in that case the fungus which can easily cause the disease, and that will be cause Shigatoka disease. Their diagnostic symptoms. Early symptoms appear on the lower leaves, initially small reddish brown specks develop on leaves near the tip of margin or lamina. Specks may also be produced near the midrib. Then the specks increase in size and turn into spindle shaped spores with reddish brown margin and grey center surrounded by a yellow halo. Yellow halo, that means like the inside the color will be yellow, and on the periphery the color will be lightish black. So this type of color will be called yellow halo. Spores formed near the midrib enlarge and extend towards the margin of lamina. Like that, here you can see the symptoms on the leaves of the banana. So it will be like that. Then the sport collions and the entire spotted area appears dried. Disease gradually progresses into upper leaves, then the infection becomes severe after bunch emergence with the entire foliage infected under favorable conditions. Fruits in bunches of infected plants are underdeveloped and may ripen prematurely. So these are the symptoms of yellow Shigatoka and black Shigatoka disease. These are the diagnostic symptoms. Here you can see that basically the symptoms of the leaves. So it will be on the margins of the leaves. Their management: planting banana in well drained soil, growing moderately resistant cultivars like Karpura, Chikrakali, planting at recommended density. There should be 1000 plant per acre. Then pruning suckers periodically avoid overcrowding in the field. Then the removal and destruction of affected leaves, which followed by spraying with Bodex mixture 1%, 1% plus linseed oil 2%. So we have to prepare a mixture with the Bodex mixture or linseed oil, and after that we have to apply it. Applying recommended dose of potassium fertilizer, because if there is the lack of the potassium fertilizer, in that case the pathogen will be very active, or if lack of potassium is there, so it will be for favorable condition for the pathogen. That's why here we are using the particular potassium fertilizer, because if the level of the potassium will be increased, then it will be manage the disease. Then spraying of mancozeb or chlorothianil 0.2% suspended in mineral or paraffin oil, spraying chlorothalonil 0.2% with non-ionic adhesive in pre-monsoon period, and propiconazole 0.1% and interspread with tridemorph 0.1% at 20 days interval in rainy period. So this is all about the Shigatoka disease of banana, yellow Shigatoka and black Shigatoka disease. Next is the Panama disease or Fusarium wilt. Panama disease or Fusarium wilt the economic impact first reported from Australia in 1874. The popular variety Gros Michael, mostly grown for export quality fruits, was most susceptible and had to be replaced with Cavendish bananas, which were resistant in the Latin American countries. First reported in Australia in 1874, the popular variety Gros Michael, mostly grown for export quality fruits, was most susceptible and had to be replaced with Cavendish bananas, which were resistant in the Latin American countries. Cultivars like Rasthali, Gros Michael, and Karupurabali cultivars are susceptible for these diseases. Four races of the pathogen are known to exist, but race 1 is mostly prevalent in India, attacking attacking silk group of cultivars. Their etiology, like Fusarium oxysporum or the Fusarium species, that is cubens. So the primary infection, which is the clay mydiospores in soil and propagules in infected suckers, and the secondary, which goes by micro and macroconidia throughout the irrigation water. So, like throughout the soil propagules or throughout the soil and throughout the water, the disease will be spread. So, here the structure of clay mydiospores, macro and microconidia. Favorable condition like saturated, poorly drained, heavy soils, cultivation of susceptible cultivars like Amritpani, and infection by burrowing nematode that is Rhodophilus similis predispose for the plants to disease. The diagnostic symptom like conspicuous symptom usually appear on 3 to 5 months old plant, although 2 to 3 months old plants are take also killed under highly favorable conditions. Symptoms initially seen in older plants in a although 2 to 3 months all plants are also killed under highly favorable conditions. Symptoms initially seen in older plants in amid then the earlier symptoms initially seen in older plants in amid the earliest symptoms are faint yellow streaks on the petiole of oldest lowermost leaves. The earliest symptoms are faint yellow streaks on the petiole of oldest, lowermost leaves. Then the affected leaves show progressive yellowing, break at the petiole, and hang down along the pseudostem. The affected leaves, which show progressive yellowing, break at the petiole and hang down along the pseudostem. So these are the symptoms like longitudinal splitting of pseudostem is very common. Light yellow to dark brown discoloration of vascular strand in pseudostem, usually the discoloration which appears first in the outer or oldest leaf sheath and extend into the inner sheaths. The fungus grows and blocks the vascular system, resulting in wilting of the plant. The vascular discoloration in cross section of rhizome appears reddish brown towards periphery, progressing into the center of ribosome. The rhizome of affected plant, which give characteristics odor of rotten fish, is infection. If infection is due to odoratum isolate of the pathogen, young suckers also develop the disease but rarely develop external symptom. So the affected plants do not produce bunches because the leaves which will be discolored, so there is no proper photosynthesis by the leaf, so the reserve or the storage food material will be not available. That's why due to the leaves, basically the production of the fruits it will be, and we can say that the bunches of the fruits are not produced. Even if they will be produced, then fruits are malformed and they will be ripened prematurely or irregularly. However, the pathogen does not infect the fruit. Like these are the symptom. Here you can see like two plants are there. Right, left hand side it is the healthy plant and right hand side infected plant. So the leaf of the plant which completely discoloration or decoloration, and they completely turn green to yellow in color, and now they will be fall down. That's why it will be directly infect to the fruit production. Life cycle of Fusarium in banana plant. Like there is formation of resting spores in drying plant, then the death of the plant, then it will be return of resting spores to soil, then germination of resting spores in soil, after that infection of young fleshy roots and wounded roots, then again they will be moved towards colonization of plant and spore formation. Then there will be the movement of xylem, and this kind they will be infect the plant. Their management, like use of disease free suckers for planting, then we have to avoid ill drained soil, flood following for 6 to 14 months, or crop rotation with puddle rice. Then application of lime is 1 to 2 kg per pit to the infected pits after chopping of the plant parts. Then the dipping of suckers in carbendazim 0.1% and solution before planting, neem cake plus trichoderma viride should be applied in planting pits. Then soil drench with 0.2% carbendazim or rhizome injection with 0.2% carbendazim. Growing resistant Cavendish varieties like Vasrai, that is Vimena kelly or dwarf cavendish, and the puan, that is Karpura, Chakrakeli, etc. Then the Moko disease or bacterial wilt. Like here, one disease which have two names, and we can call the disease like Moko disease or bacterial wilt. Their economic impact first recorded in Guyana in 1840 in Moko plantain. So there is the reductions in yield due to MOCO of up to 74% have been reported in Guyana. A disease outbreak in Trinidad in the late 1890s caused severe losses. So, a disease outbreak in Trinidad in the late 1890s caused severe losses of Moco cooking bananas. Moco is endemic to Central and South America, not reported from India. Moco disease affect ornamental, that is Helioconi species. It also affects commonly grown desert bananas. So it also affects commonly grown desert like bananas, Musa, plantains, Musa paradisica, and cooking bananas. Their etiology, like raw Listonia solanicidum, raised to biova, one pathogen is spread through infected plant parts, including root, trunk. So it can be spread through infested soil on hands, tools, shoes, machinery, animals, and water farm off. Favorable conditions like crop residue infected with pathogen in the field, injured roots which caused by farm tools or by the soil paste, crop residue which infected with pathogen in the field, injured root caused by farm tools or by soil paste, then warm temperature and high soil moisture, then the slightly acidic soil, poor and unfertile soil, and infestation by nematodes. So these all are the conditions which will be favorable, and in these conditions basically the pathogen which can cause the disease easily. Their diagnostic symptoms. Symptoms start on rapidly growing young plants. The youngest 3 to 4 leaves turn pale. The youngest 3 to 4 leaves turn pale green or yellow and collapse near the junction of lamina and petiole. Then the characteristics discoloration of vascular strain, wilting and blackening of suckers, characteristics discoloration of vascular strain, wilting and blackening of suckers, Vascular discoloration, like pale yellow to dark brown or bluish black, is concentrated near the center of the pseudostem, becoming less apparent on the periphery. So there is the grayish brown bacterial ooze is seen when the pseudostem of affected plant is cut transversely, and firm brown dry rot is found. Then a firm brown dry rot is found within fruits of infected plants, and then the death of whole plant occurs under severe infection. So these are the symptom of the disease, that is the wilt disease. Okay. So the complete plant leaves, those are wilt and they are completely dry, and after dry they will be fall down automatically. That's why they will be also decrease the production. Like inside, this type of symptoms will be appear whenever we will cut the section. These are their management. Grow relatively resistant varieties like Puan and Monthan. Adopt strict plant quarantine and photosanitary, which measures and plant healthy suckers. Exposure of soil to sunlight during dry hot weather. Then eradicate infected plants and suckers by rotting or killing in situ by application of herbicide. So there is the disinfestation of tools with formaldehyde, which diluted with water in 1:3. Like formaldehyde only like 1%, then the water should be thrice. Okay. Then the crop rotation, 3-year rotation with sugarcane or rice, and providing good drainage. So allow fallow period or flooding during off season. Fumigation of infected site with methyl bromide or chloropicrin. Drenching soil. Fumigation of infected site with methyl bromide or chloropicrin. Drenching soil in infected pockets with bleaching powder solution 1.5% and the Bordeaux mixture, that is 1%, and streptocycline 0.2%, and the biocontrol with the Pseudomonas fluorescens. Okay, thank you. Here is on root or soft root or tip over disease. Their economic impact, first recorded in Honduras on Gross Michael in 1949, Israel, Central America, Jamaica, Papua New Guinea and India. Reductions in yield due to tip over of up to 93% have been reported in Guatemala. Evidence is up to 70%. The disease has been reported on Nandaran and dwarf Cavendish banana in Kerala, Tamil Nadu, Karnataka and Gujarat. Etiology: Irvinia caratura subspecies Irvinia chrysanthemi. Primary spread: bacterial cells in soil and through diseased plant suckers used for planting. Secondary: bacterial cells throughout the irrigation water. Favorable conditions: juvenile stage of the crop is highly susceptible 3 to 4 months after planting. Prevalence of high temperature 37 degrees centigrade in early stages of crop growth. Waterlogging and high temperatures favor infection by Irvinia chrysanthemi. Growing susceptible cultivars like Cavendish and Tela Chakarkeli. Usually young plants are attacked under favorable conditions. Even mature plants are infected. In newly planted young sprouting sucker, the rhizome is infected and infection progresses into pseudostem, leading to toppling of tip over of the germinating suckers, shock, spore develop on outer leaf sheaths on base of pseudostem near the soil line. The spores turn brown and soft rotting of pseudostem tissue and rhizomes takes place. Infected plants have scanty roots with dark brown lesions and necrotic tip. Infection is spread to upper part of pseudostem and to rhizome. In many cases, the pseudostem tips over because of breaking caused by rotting at the ground level. Mature plants are slow to exhibit symptoms and may produce small-sized bunches which may fail to emerge from the shoot tip. In infected rhizomes, pockets of dark water, soaked areas develop. Infection may result in the production of cavities which resemble root borer tunnels. Diagnostic symptoms are: these are the diagnostic symptoms. Management: avoid planting susceptible varieties during periods of high rainfall or in waterlogged soils in problematic areas. Crop rotation with rice or sugarcane for 3 to 4 years. Ensure that only healthy suckers are planted. Dip the suckers in copper oxychloride 5 gram per liter plus monoprotophos 2.5 ml per liter solution. Drenching the base of the plant with Pseudomonas fluorescens 1 2-liter plant 50 gram per liter of water at 0, 2nd, 4th and 6th month after planting. Maintain proper soil aeration by intercultivation, then maintain optimum soil moisture by light and frequent irrigation, grow sunhemp or cowpea 2 to 3 times in interpieces till 5 months after planting, then remove the destroy infected plants along with roots. Drench the infected suckers with bleaching powder 1.5% solution. Restrict to one or two red rooms based on disease incidence. Drench the infected suckers with bleaching powder 1.5% solution. Then restrict to one or two ratoons based on disease incidence. Then bunchy top, curly top, cabbage top, and strangles disease. First reported from Fiji in 1889 in Cavendish varieties. Around 1940, introduced into India from Sri Lanka through cyclone. The virus is a domestic quarantine based India around 1940, introduced into India from Sri Lanka through the cyclone. The movement of planting suckers from northeast Tamil Nadu, Karnataka, and Kerala to other parts of the country is restricted. Their etiology, then banana punchy top virus, primary virus particles, throat disease suckers used for planting, secondary virus particle transmitted by banana black aphids, Pantalonia negronerosa, then Colocasia esculenta serves as a talent reservoir host. Progressively increasing temperature from February onwards favors virus spread and symptoms expression. Then prevalence of infected reservoir host, that is Cana, Heliconia, and Esterlitzia. Prevalence of aphid vector. Infection may start any stage of crop growth, then paling of lamina with intervenial chlorosis that is evident against light, prominent dark green streak on the petioles and is evident against light, prominent dark green streaks on the petioles and the long midrib and leaf veins that range from a series of dark green dots to a continuous dark green line, that is Morse code. Infected plants show marked stunning. Diagnostic symptom: leaves are reduced in size with narrowed lamina and shortened petiole, reduced at shortened internodal length, become erect and brittle and crowded at the top. produced at shortened internal length, become erect and brittle, and crowded at the top, bunchy top. Leaf margins also show chlorosis and slight curling and necrosis. Infection of young plants leads to failure of bunch emergence. Then become erect, brittle and crowded at the top. Leaf margin also show chlorosis and slight curling and necrosis. Infection of young plants leads to failure of bunch emergence and tips of inflorescence. Bracts of infected plants remain green and do not turn to normal pink or purple. So these are the symptoms: virus infected or healthy leaf, virus infected green J hooks and Morse code. Adoption their management, adoption of strict quarantine measures occurs, or tissue culture plants for planting, periodical monitoring and regular herbicide 2,4-D. Raising barrier crops like sunhemp in 3 to 4 rows on the field boundaries to check aphids from entering the field from neighboring infected fields. Vector control with systemic insecticides, phosphomidone 1 ml per liter or methyl, dimetone or dimethoate 2 ml per liter, then discouraging intercropping with colocasia in disease endemic areas. Thank you.